There's a difference between biochemistry and molecular biology. I always prided myself in being a biochemist who studies the chemical properties of biomolecules and sometimes molecular pathways. Molecular biology is a little different. We're talking about macromolecules that help the cell function in a way that allows it to live and continue through its life cycle. So I wanted to go back to the basics and really understand molecular biology for all it is. I took one course in molecular biology in grad school, and I'm going back to review the textbook from that book, which I actually never read in its entirety. But now I'm going through page by page and picking up all the details that I wasn't able to the first time. It's probably going to take at least 10 episodes to cover the whole book, and I'm not entirely sure I'm going to get through it, but that's my intent for now. Today, I'm just going to briefly touch on some general concepts in genetics and also touch on replication, transcription, and translation, and some cloning techniques. Gregor Mendel showed in 1865 that parents have 2 copies of each gene, and that each cell is a diploid cell. We know that each gene can have different versions of that gene. They're mostly the same, but they have slight differences that can have big impacts. And I mean slight differences in the DNA sequence of each of those genes. One version of the gene is called an allele, and there can be many different alleles for a given gene because there can be different types of mutations in that gene. There are many alleles possible, but typically in humans, we often have single nucleotide differences or single nucleotide polymorphisms, SNPs, that cause us to have different alleles for certain genes. Because we have 2 copies of each gene, we know that each gene can be the same, homozygous, or they can be different alleles, heterozygous. One version of the gene can be dominant, meaning that its phenotype will be manifested when the other copy is recessive. So for a given gene, you're either homozygous or heterozygous, dominant or recessive, and you can use the language heterozygous dominant or homozygous recessive for a gene. Sex cells are interesting because their chromosomes or their genome is haploid. They're haploid cells, meaning there's just one copy of each chromosome. A certain trait for an organism can be sex-linked, meaning it is passed down through the sex chromosome. Recombination that happens during meiosis can often prevent sexually linked genes from always being linked to the parental chromosome. So the offspring may have a sex chromosome that is partly from the mother and partly from the father. This is kind of embarrassing, but I forgot how many chromosomes we have in our genome. Turns out we have 22 autosomal chromosomes, which are all the chromosomes besides the sex chromosome. And there is only 1 pair of sex chromosomes. So 22 pair of autosomal, 1 pair of sex chromosome. The location of each gene on the chromosome has a locus. The location of a gene on a chromosome is its locus. The DNA itself in a chromosome is mostly in the B form. That's like the standard form. But sometimes it does form an A form, which is also a right-handed helix, like our standard B form. But the bases aren't fully parallel to each other. They sort of are bent at an angle. There's a third form of DNA, which is called zDNA, and this happens with their alternating GC base pairs. And this form of DNA is really cool because it's left-handed. DNA is often measured by its UV absorbance at 260 nanometers. When it's in its double-stranded form, it has a specific absorbance that you can use to quantify the amount of DNA you have present. But when you heat up that sample, the DNA strands become separated, they're denatured, and a hyperchromic shift occurs, which means that the absorbance at 260 nanometers increases because the quenching that happens in the double-stranded form is taken away because those strands are separated from each other. When you allow single-stranded DNA to anneal to itself, you're forming a hybrid. And recently I was thinking that this might be a misnomer because a hybrid, in my mind, means the coming together of 2 different things. And in my mind, that was DNA and RNA coming together to form a hybrid. But in fact, in the Weaver textbook, a DNA hybrid can exist where 2 single-stranded DNA come together from different origins. to form a hybrid. The DNA content of a cell can be expressed by its C value, which is the amount of DNA in picograms in a cell. And this doesn't necessarily reflect the complexity of the organism because we don't know how many genes that genome might have versus a number of introns that aren't used for protein synthesis or RNA synthesis. For example, there are some plants that have a much higher C value than our genome, over 100 times more. There are also plants that seemingly have similar functionality but have less genetic material than we do. So it's not necessarily a reflection on the abilities of an organism. Obviously, we have a higher consciousness than plants, but not necessarily a higher C value than some plants. The one thing I will say on replication is that when our cells divide and our genetic material has to be duplicated, we use a semi-conservative mechanism, which means that the parental strand is conserved in each of the daughter helices. This isn't true for all organisms. There are some bacteria that replicate using rolling circle amplification and just make multiple copies of a single strand of that DNA. It's not symmetrical and it's not semi-conservative because only one of the strands is replicated. When DNA is converted to RNA, in the process of being converted to protein, transcription occurs. Transcription relies on an RNA polymerase that binds to a promoter of the DNA in the genome. During this initiation, there is an initiation factor bound to RNA and is different in different organisms. But what happens during this initiation is 12 base pairs. Around 12 base pairs is melted, and the first initiating nucleotide triphosphate that comes in is usually a purine and comes in and base pairs with its complementary base near the promoter. And it's really interesting that transcription does not need a promoter for this, like replication. This is something that didn't really sink in the first time I took molecular biology, or maybe I just forgot this concept. And I think that's so cool that it can initiate this RNA polymerization without a primer. So once it elongates, this is an asymmetrical process because only one strand is transcribed. There is a termination where the RNA is loosened from the DNA and it floats away, and that transcript is then potentially translated by a ribosome. The ribosome will have a large subunit and a small subunit. The small subunit is often referred to as a 30S and the large is a 50S according to its sedimentation coefficient, which is a measure of how large the biomolecule is as a result of its ability to travel through a sucrose gradient in an ultracentrifuge. Actually, I think it's a cesium chloride gradient in an ultracentrifuge. In the small subunit, there's a ribosomal component which mutates very slowly, and this portion of the ribosomal RNA is used to identify different organisms because it uniquely identifies a species. Once the RNA binds the ribosome, and I'm going to go into this in more detail when I study translation later in the textbook, but the AUG codon is used as the initiating codon. In bacteria, the start codon also has a Shine-Dalgarno sequence that's upstream of the start codon. That helps the ribosome recognize that this is a start codon so that it can begin translation. Eukaryotes work a little differently. They have a methylated nucleotide cap that is recognized by an initiating factor that allows for initiation of translation with the ribosome. The 5' untranslated region of RNA is often called the leader, and the 3' untranslated region of the RNA is often called a trailer. The poly A tail that we often hear about in mRNA is actually not encoded in the genome. In eukaryotes, it's actually appended to the 3' end. After the 3' end of the mRNA gets cleaved, a poly A, this is downstream of the stop codon, a poly A tail gets put on the 3' end and helps protect the mRNA from degradation. Sometimes DNA gets mutated, which means that like one of the nucleotides gets replaced with a different nucleotide, and this can be due to a polymerase error. But sometimes that change doesn't have any impact on the amino acid sequence because the codons are degenerate, meaning that there are multiple letters that can encode for the same amino acid. And when this happens, when there's no impact on the amino acid, this is called a silent mutation. But sometimes amino acid is changed to a similar amino acid. This is called a conservative mutation. There are enzymes called restriction internucleases that chew up DNA at specific sequences that are palindromic. And the way that these internucleases can exist in a bacteria without chewing up much of the DNA and destroying itself is the internucleases are also paired with methylases that recognize the same sequences that the restriction internucleases recognize and cleave. And these methylases protect the DNA from cleavage. Even when the cell is replicating, the daughter strand that is being formed during replication isn't methylated yet, but the parental strand has a methylation, meaning that the helix is hemimethylated at that position. And this is enough to protect the genome from being cleaved by a restriction enzyme. If you want to transfer DNA into a cell, sometimes you use a plasmid. And in order for this plasmid that is circularized, which can be 5,000 or 10,000 base pairs, the plasmid needs to have an origin of replication so that it is replicated once it enters the cell using either electroporation or a calcium treatment. When you're using a phage vector to introduce DNA to a cell, you often, instead of getting colonies of bacteria on your plate, your goal is to actually get a hole in a lawn of bacteria because that phage vector is entering the cell and it is forming phages and lysing your cell, forming a hole in your lawn of bacteria. Lambda phages are really cool because they can transport more DNA into your cell because plasmids have kind of a limit in the amount that they can transport using a calcium heat shock or electroporation. They can transport 12 to 20 kb of DNA into lambda phage in addition to the arms, which are extra DNA that encode for everything that's required for the production of the phage molecules inside the cell so that it can assemble phage molecules that will then lyse the cell and infect other cells. This type of cloning can be useful to generate genomic libraries. Lambda phage vectors are also known as replacement vectors, as the DNA in the phage is replaced with new DNA. Plaque hybridization is a method to determine which holes in your lawn are formed from a phage that has a particular gene in it. It can trace genes to phage plaques. So this is like the holes in the bacterial lawn are called plaques. Another type of cloning uses cosmids. Cosmids are longer stretches of DNA that can be bundled up into a phage, but it doesn't have the arms that contain all of the information for producing the phage. So what is done is you basically package the cosmid DNA into a lambda because it has these COS sites in the end, these cohesive sites. But this cosmid has an origin of replication. So once the phage is assembled and the cosmid DNA is transported into a cell, it then is replicated in that cell using the origin of replication, like a plasmid would, but it can't infect other cells. It basically can divide and be replicated and present in those cells. Another type of cloning uses an M13 bacteriophage, which is just a different type of phage. This phage has a single-stranded genome, which makes it easy to manipulate using site-directed mutagenesis. I hope this video wasn't too scattered for you. I covered a lot of things, but these are all things that I didn't know before and I think are really interesting. So I hope you got some value out of it as well. Have a great week and stay tuned for the next video. Take care.